In parenteral, or injectable, drug manufacturing, there is a dangerous paradox. Killing a bacterial contaminant does not make the product safe. Early 20th century intravenous drugs like salvarsan frequently caused a severe reaction known as injection fever. Doctors quickly realized that the solutions required absolute purity, not just an absence of living microbes. The culprit behind injection fever is the bacterial endotoxin. Endotoxins are amphiphilic lipopolysaccharides, or LPS, which are large molecules located in the outer membrane of gram-negative bacteria. LPS is incredibly resilient. These molecular fragments survive extreme heat sterilization and easily pass right through the standard 0.22 micron aseptic filters used to purify liquid drugs. Because they are hydrophilic, LPS molecules also have a high affinity for adhering to the plastics and glassware used throughout a manufacturing line. If these lingering fragments are introduced directly into the human bloodstream or cerebrospinal fluid, the immune system mounts a massive, uncontrolled defense. This animated diagram shows exactly how that reaction begins. The lipid portion of the endotoxin binds to a specific receptor called toll-like receptor 4, or TLR4, on the surface of human macrophages, triggering a severe innate immune cytokine cascade. Because endotoxins survive the very processes designed to ensure sterility, specialized pyrogen detection is a non-negotiable requirement to prevent fatal septic shock in patients. Historically, the pharmaceutical industry relied almost entirely on end-of-pipe batch release testing, pulling a sample at the very end of the line to catch endotoxin contamination. Today, agencies like the FDA demand continuous oversight rather than reactive testing. Regulatory frameworks, specifically the ICH-Q9 guidelines, mandate quality risk management, or QRM. QRM is a systematic process for the assessment, control, communication, and review of risks to the quality of the drug product across its lifespan. This aligns with the philosophy of quality by design, which dictates that endotoxin control cannot be tested into a product at the end. It must be engineered into every step of the manufacturing lifecycle. True patient safety requires a vertically integrated risk map that spans the entire manufacturing pipeline and reaches all the way down into the global reagent supply chain. On a sterile facility floor, the primary vectors for endotoxin ingress are incoming raw materials, excipients, and the personnel handling the equipment. The highest risk critical control point, or CCP, in the entire manufacturing pipeline is the water for injection system. A CCP is a specific step where control can be applied to prevent or eliminate a pharmaceutical safety hazard. These highly purified water distribution loops are inherently vulnerable due to areas of potential stagnation, temperature fluctuations, and dead legs, sections of piping geometry where water does not actively flow. Inside those stagnant zones, microbial biofilms form. The bacteria cling to the steel pipe walls and continuously shed LPS molecules into the fast-flowing water stream. Manufacturers map these microbial control risks across upstream, formulation, and fill-finish operations using process hazard analysis. Process hazard analysis is a structured, team-based method used to identify and evaluate vulnerabilities in complex industrial processes. Historical environmental monitoring data pinpoints aseptic filtration assemblies and container closure systems as highly vulnerable control points. Securing these points requires rigorous physical mitigation strategies, such as executing validated cleaning-in-place cycles and physically redesigning piping to eliminate dead legs entirely. Even the most highly purified water systems act as continuous, invisible endotoxin generators unless they are continuously monitored and geometrically optimized. Even with strict physical controls, detection can fail due to a dangerous biochemical anomaly known as low endotoxin recovery, or LER. This diagram illustrates the mechanism of LER. Specific drug formulations, particularly complex protein and surfactant matrices, chemically mask the endotoxins, physically blocking the reactive lipid domain from being detected by standard assays. LER produces false negative results, meaning potentially lethal products can easily pass final quality control checks. To counter this, manufacturers use risk assessment flowcharts like this one. It maps out the integration of personnel hygiene training, historical bioburden data, and supplier performance scoring to create a holistic defense. Regulators also mandate hold time studies. These tests prove that endotoxins can still be accurately recovered and detected throughout the exact storage conditions and specific duration of a product shelf life. A negative test result on a complex biologic does not guarantee safety. It may simply mean the pyrogen has been biochemically camouflaged. Beyond factory floor anomalies and chemical masking, there is a systemic vulnerability in testing reagents themselves. During World War II, the massive demand for intravenous fluids on the battlefield catalyzed the need for a new pyrogen test. It had to be faster and more efficient than injecting live rabbits and monitoring their temperatures. In the mid-20th century, researchers discovered that amoebocytes, specialized blood cells from the horseshoe crab, would rapidly clot in the presence of endotoxins. That discovery created a massive supply chain vulnerability. Today, the global pharmaceutical industry relies almost entirely on the blood of this single wild-harvested marine species to run the limulus amoebocyte lysate, or LAL, test. Sole sourcing of vital quality control reagent from nature introduces strict ecological constraints and leaves the industry highly vulnerable to regional demand shocks. As global pharmaceutical trade expands, the demand for sterile testing continues to outstrip biological limits of the horseshoe crab population. The safety of our most cutting-edge biopharmaceuticals is precariously tethered to the fragile ecological limits of a single crustacean. To eliminate this ecological dependency, the industry is turning to a genetically engineered alternative called recombinant factor C, or RFC. This diagram compares the two methods. Left, the multi-step enzymatic cascade of the traditional LAL test. Right, the synthetic RFC pathway perfectly mimics the initial LAL trigger. It outputs a precise, quantifiable fluorescence peak, offering equivalent or superior sensitivity compared to the qualitative clot. Global regulatory bodies are coordinating the adoption of this technology through harmonization efforts like the ICH Q4B Annex 14 guideline. Major pharmacopoeias, including the US, European, and Japanese organizations, are formally integrating RFC into their testing standards to ensure supply chain resilience. Recombinant technologies decouple patient safety from ecological limits and stabilize the global medical supply chain. Both LAL and RFC share a critical blind spot. They are only sensitive to endotoxins from gram-negative bacteria. They cannot detect non-endotoxin pyrogens, or 𝗫s. 𝗫s include cell wall components from gram-positive bacteria and pyrogens derived from viruses and fungi. To capture all pyrogenic threats, laboratories use the monocyte activation test, or MAT. MAT is an in vitro assay that measures the release of cytokines from human blood cells when exposed to pyrogens. This diagram shows the MAT mechanism in action. Human monocyte cells react to a diverse mix of both 𝗫s and endotoxins by releasing a measurable cloud of cytokine markers, accurately simulating a true human immune response. While RFC solves the supply chain fragility of animal-based testing, MAT closes the biological safety gap by providing a truly predictive human model for all classes of pyrogenic threats. Endotoxin control has evolved from a simple laboratory release test into a systemic engineering and supply chain imperative. It requires precise physical execution on the manufacturing floor, utilizing seamless dead leg-free piping, advanced real-time sensors, and validated cleaning cycles. It also demands analytical evolution, shifting away from reactive, animal-dependent LA assays toward proactive synthetic RFC testing and comprehensive MAT frameworks. By engineering out vulnerabilities at every level, from pipe biofilms to reagent limits, the pharmaceutical industry secures the delivery of safe biologics to patients worldwide.